Welcome to the Molecular Therapy Podcast. I'm Dr. Rory Bricker-Anthony, Scientific Editor of the Molecular Therapy family of journals. This episode is focused on the Molecular Therapy Advances upcoming special issue, Beyond Prevalence, How Rare Diseases Are Reshaping Therapeutic Innovation. I'm here today at the 2026 ASGCT Annual Meeting in Boston with the Editor-in-Chief of Molecular Therapy Advances, Dr. Mohammed Abu Elanin, and the guest editors of the special issue, Dr. Jennifer Adair, Dr. Claire Booth, Dr. Terry Plott, and Dr. Donald Cohn. Before we get started, join leading policymakers, industry executives, and scientific stakeholders at the ASGCT Policy Summit, a one-day event in Washington, D.C., on Monday, September 14th, 2026. Be there to discuss the most pressing issues in the field and connect with leaders working across regulatory, manufacturing, access, and innovation in CGT. Virtual registration is also available. Visit asgct.org to learn more. And now I'll hand it off to Mohammed and our guest. Welcome.
Thank you. This is a very exciting podcast, and we have top leaders in the field. Let me ask you, how did you get into rare diseases as a field? And do you feel like the field has matured, or we still a long way to go?
Yeah, so I'm a pediatric immunologist by training, and I wanted to go into pediatrics, and I really enjoyed doing transplant and immunology, and just by virtue, pretty much every immunological disease is like a 1 in a million or 1 in 100,000, so I just ended up in a rare disease field. That's how I ended up in it. And has it matured over the years? Yeah, absolutely. I've been working in the field probably for around 20 or so years now. And I was lucky enough, in this area in immunology, it really has been, I would say, one of the foundational test beds of all gene therapies. The first gene therapies done 30 years ago. So when we look back at what was done then and the sort of things that we're doing now, we've learned from all of that. And we've got new technologies, a huge range of diseases that we can treat now, different kinds of cells that we're addressing with it. So yeah, I'm saying it's moved on.
Thank you, Terry. Please.
Yeah, so I'm also a pediatrician. I'm a pediatric pulmonary physician. So I got into the field because the Cystic Fibrosis Foundation both funded my research training and got me on track of trying to develop the AAV system initially for cystic fibrosis. What we found out as we went through that and brought it through to the clinic was that the vector system was probably best suited for single gene disorders in non-dividing cells, such as in the central nervous system, retina, cochlea in the ear, and so forth. So I've followed the path of where the vector's been helpful, so that we could connect with other disease populations. But it really all kind of, for me, motivationally tracks back to being a pediatrician and trying to work with families who have these different conditions. As far as the field maturing, the AAV part of the field, certainly when we did the first AAV human trials in the mid-90s, we had very small doses we were able to manufacture, and we really had limitations. We didn't have many different versions of AAV, different serotypes to target different cells. And so what we're seeing now is that different variations of AAV are really finding their own place in the clinical armamentarium overall. And so that is partially matured, I'd say. But the number of diseases, the number of patients who could potentially benefit is still far greater than the number who are benefiting. So we have a long way to go.
Yeah. Thank you. Jen, please.
I would say for me it was a little bit more just time of life. So I call myself a child of the Human Genome Project, and I will be the only non-MD, I think, on the podcast as a guest editor. I started my PhD in 2001, and the first human genome was solved in 2003. So I think it was natural that human genetics was going to play a big part. I think as the field of gene therapy progressed early on, it was the untreatable diseases, or the diseases that traditional disease-modifying medications were not able to actually make a dent in, that were really the first candidates, and those primarily represent the rare disease population. I knew when I finished my PhD that I really wanted to do translational science. I wanted to be more connected to taking what was happening on the bench into what was happening in patients, but I didn't have the clinician's degree to do that. And so I found a laboratory that was translating a phase 1 clinical trial of blood stem cell gene therapy that wanted someone who could understand the science to do some of the regulatory work and some of the process development work. And then that ended up being for Fanconi's anemia, which was a DNA repair disorder. And that patient population strongly advocated. The Fanconi Anemia Research Foundation and the families fighting for patients with Fanconi anemia presented me with this phrase that I carry with me through my career, which is, if you can solve the problems that you need to solve to treat the hardest to treat patient, then you've really solved the problems that you need to treat everyone else. And so I feel like that was really inspirational to me in my career. My career now balances between diseases of global health burden and also rare diseases, but inspired by those families and the rare diseases involving DNA repair.
Amazing, thank you. Don, please.
Yeah, I'm like Claire. I'm a pediatric bone marrow transplant physician, and so a lot of the patients we see, other those with leukemia, are immune deficiency patients or other inborn errors of metabolism, all of which are rare. And so those are just the diseases that we've always studied. So as we started working on gene therapy, those were the diseases we decided to apply ourselves to. So I don't think I intentionally thought I was going to work on rare diseases. I thought I was going to work on the diseases of the patients that I saw a lot of. I think over the last decade or so, I think the field as a whole has recognized this issue of rare diseases and how neglected they've been. And so I think it's really great, the new recognition of that and all the ways things are coming together with patient advocacy groups and others to really advocate for those patients and develop these therapies. As we'll probably discuss further, they represent challenges because they're not commercially viable, or they haven't been, and so that's the challenge of how to translate these great therapies to a sustainable therapy that's available for patients, hopefully for generations to come.
Amazing, thank you. For me, most of the work that you have done has been the foundation of what we see now in patients. And Jen, I think you deserve an honorable MD for the amazing translational work that you have been doing. Maybe I can follow up with Don and Claire, because you have been working on the ADAS kit, and it has actually matured enough to see a long follow-up in patients that have been treated. What kind of carry-over lessons to other rare diseases that you think we can take, and what comes next for ADAS kit?
Well, so Claire will be speaking about this. We sort of are taking different approaches. We worked in parallel on the same approach, the same vector. We even shared the batches of vector. It got licensed to a company that was going to take it to FDA approval at least, and then they decided because of commercial concerns not to do that. So it came back to us about 3-4 years ago, and we're both pursuing paths to try and make it sustainably available. So we've actually started a company, Rarity Public Benefit Corporation, that will try to get it FDA approved. And what the burden of data of manufacturing primarily to get FDA approval that you can make the drug consistently at high quality is a lot, and it's very expensive. And so the company is trying to deal with that we have some grants from the California Institute for Regenerative Medicine and some investments so far to try and support doing that and bring it to commercialization. Claire is taking a different approach. Maybe you want to talk about that.
Yeah, I'm just going to jump back to the first thing that you said though about what can we carry over, and I think I'm just going to say that I think that lentivirus is great, and it's a technology that we've learned so much from. We have huge amounts of follow-up. It's a de-risked strategy. So whilst I'm as excited as the next person about all the editing platforms that are coming out, I will carry over for the lenti work that we've done for ADA and other things. Yeah, it's a great platform to treat some of these rare diseases. But Yeah, we're taking a different kind of perspective in the UK because we have a really different regulatory framework and a different payer framework, and that actually is to our benefit in some ways. So I work at Great Ormond Street Hospital, and we got some funding from Gosh Charity and LifeArc to really do a scoping exercise to see whether we could, as an NHS trust hospital, become a license holder for a rare disease advanced therapy and deliver that within the NHS kind of framework. And whilst we're using ADA SCID as a kind of example to see how we may be able to do this, we're really hoping to develop a pipeline for academically developed therapies for rare diseases that are just not commercially feasible. So we're pretty lucky that we have our own academic GMP facility, so we're not beholden to a CDMO or that kind of expense on that side. So we're looking to how can we make our facility commercialized, and then just sort of working through all of the regulatory submissions, talking about patient access with the payers, and then really trying to put some sort of business modeling around it so that if this works, we can do it in a sustainable fashion for things coming through in the future.
And I think this is a very critical point about access and reimbursement that we will touch on in a minute. Before that, I would like to go back to Terry, and you did talk about AAV, and AAV has become a standard vector for many gene therapies. And you did mention some of the advances, but maybe you can also talk about where does it fall short and what comes next in terms of improvement in AAV.
Yeah, when I think about it, I think there are two kind of types of limitations. One is inherent in the therapy itself, which is that we are now reaching a point of dose-limiting toxicity with AAVs, particularly when given at high doses by systemic IV administration. When we're trying to, by more or less brute force, treat a lot of different cells throughout the body, such as the entire musculature, heart and diaphragm included in patients with Duchenne muscular dystrophy, the doses we end up using are extraordinarily high, and we are seeing complications from that in some cases are very severe and in rare cases are fatal. So we're up against that biology, which I think we need to go back and again study more mechanistically. But I think we're also, with AAV, up against the same hurdles that Claire talked about for treatment of immune deficiencies, which is that we need innovation to spread the technology, the access to the technology. And I think the innovation, we can benefit from innovation in manufacturing if it either results in a less expensive manufacturing or in the case of AAV, a lower dose being needed. That could certainly lower the necessary cost. We need innovation in terms of regulatory innovation, treating things more as platforms, and also looking at alternatives to approval rather than going through an entire de novo process with each iteration or different vector. And I think we also need innovation, as Claire already mentioned, in the payer frameworks, because our payer frameworks are best suited for one who takes a pill every day, there's an ongoing, lifelong purchase of the therapeutic. We're not really well set up for a very expensive therapeutic that has the potential to treat somebody for many, many years or a lifetime. And so while many people have thought about that, the real world application of that, particularly when the individual therapeutic only is for a small population of people, I think that's going to require some innovation from economists. And I think, a number of, Jen in particular, have been working on modeling this for both low middle income countries and in the U.S. So I think, like a lot of problems, it drives innovation.
Perfect. I love how the discussion is taking us to access, and maybe, Jen, you mentioned the global impact of what you're doing and the public health impact. And as we know, sickle cell disease hits harder in low and middle income countries. So how can, talking about access and innovation in manufacturing, innovation in reimbursement, regulation, so how can we make sure that these people get these treatments that they need the most?
Yeah, so I think, sickle cell is a great case study. was the first molecular disease actually identified in humans. It is a disease of global health burden. It's nearing 10 million patients globally, but the vast majority reside in areas where malaria is endemic because the mutation naturally selects in those populations. It's a point mutation, so it's the same point mutation in every patient with that disease. But because it primarily impacts poorer nations and generally people of color, it is subject to all of the systemic barriers that any treatment faces. It is important to note that it's not just about focusing on transformative medicines and access. The population in general has not had significant effort put into even just disease-modifying medication development until the last 2 or 3 decades. For many, many years, there was only one repurposed chemotherapy available to treat these patients. There is a lot to do in this space. Even in the U.S., more than half of the patients diagnosed with sickle cell are going to be on Medicare or Medicaid. So they're going to rely on government-funded healthcare in that capacity. And so that creates all of the challenges. We have multiple strategies from a gene therapy perspective to combat the underlying biology of that single point mutation. Most of them work, but the standard process of selecting winners in authorization in the US resulted in 2 therapies getting approved, which was a huge success and a tremendous amount of work for the academic medical centers that developed those approaches and the commercial entities that took those approaches on to get them to authorization. But it also resulted in this competition where the quote-unquote losers, those who didn't make it to that finish line, abandoned, despite having positive safety and efficacy data. And we're missing an opportunity right now to transfer some of those technologies to the places where they could work most, to learn as much as we can, to bring populations along with us. We often think, it's innovation in designing the next shot in the arm that's actually going to simplify delivery, but we're asking that to go into populations that haven't been brought on the journey of cell and gene therapy development at all. And fundamentally, that comes back to what we know about human genetics. It's just an unfortunate reality that the vast majority of the human genome sequences, I go back to my being a child of the human genome, the vast majority of the human genome sequences we have are not actually representative of the true diversity of the human population. And so we're going to face new challenges in the field if we don't start bringing underrepresented human genomes into the development process, and disparate populations who have different healthcare systems and different ecological domains in which they reside into the conversation, and invite them to be partners at the table in developing what is actually, I think, going to be probably the most broadly globally applicable treatments in the next generation.
I just want to say that I think the efforts that Jennifer's making are just amazing to take it beyond our little environment and reach out to the world with these therapies. So it's really admirable that you're taking this on and being a leader in the area.
Well, and I need to say thanks to folks like you guys who have developed therapies that actually work in patients that give us an argument to make. I can't say enough about how important it was to have transformative treatments that combat the underlying biology. We say in the sickle cell space a lot, once those authorizations were announced and people heard about gene therapy on the news, they started coming to clinic in new ways. There's something different about being diagnosed with a disease for which you have limited or no treatment options versus having a disease where there's now potentially something that's at least functionally curative or very disease transforming on the table. And it creates this conversation that we failed to have in a lot of spheres around. We need to have diagnosis and screening. We need to have disease-modifying care to make sure patients are ready for that next generation of treatments that are truly going to be transformative.
I also believe representation in clinical trials is very underestimated. And if I take this argument to rare diseases where we have a small patient population and very difficult trial design to build a strong evidence base. So maybe for Dawn and Claire, you can tell us more how you overcome recruitment and trial design for these patients, knowing that you have very few patients to treat, and the FDA would usually require high level of evidence.
Well, I think for ADA, it's the first human disease to be approached by gene therapy in 1990. So it's been around for a while, and as it's gotten more effective in the field of the physicians who see these patients initially, clinical immunologists, bone marrow transplant physicians, it's now recognized as the best treatment. I think pretty much any physician who cares for these patients would like their child to get, for ADA SCID, at least in other forms of SCID, to get gene therapy because it's so much simpler and less risky than an allogeneic transplant. And so we don't have to look for patients, they find us. And the problem is making it available and having the availability of the therapy, and that means until there's commercialization, is having grants. We've done everything to the present time on grant funding. And once you've proven something really works, it's no longer a research question. So we're sort of at that paradoxical place right now where we have something that works, but we can't give it because we don't have the funding. It's not approved yet.
Claire, would you like to add?
Yeah, I mean, I think as part of the whole regulatory innovation landscape, trial design is something that is becoming almost its own discipline. And thinking about that in the context of rare diseases, I think we're going to really benefit from that. So there are, trial designs now that just we hadn't even really thought about 10 years ago or so. So, people talk a lot now about umbrella trials and basket trials for rare diseases, and I think that's becoming a reality. And regulators are getting kind of more aware and more familiar with these kinds of designs, and certainly in the UK even suggesting them for some things. And adaptive trial designs, it's like a science in its own right now. And I think approaching it in that kind of way will really help us in the rare disease field, especially if these things are accepted by the regulatory agencies as being valid in small patient populations.
And with more outreach programs, we hopefully can have patients from across the world to receive and get enrolled in these trials so they can receive the treatments.
I guess if I could just make the point, I think often these therapies are developed at academic medical centers where for phase 1 we don't need to have validated assays and lots of other things. And it's making the leap from the academic center to the commercial where you now have to invest in the US at least millions of dollars to validate your assays to get GMP compliant manufacturing at a really high level. That's the barrier. And so I think an example, Rocket Pharma just got FDA approved for a lentiviral gene therapy for leukocyte adhesion deficiency type 1. And they, from the very beginning, worked with commercial companies. So they did everything. They had the financial resources to do everything in the professional, commercial grade from the beginning. So they were able to relatively quickly get to FDA approval. And so it's really a problem that we developed at academic medical centers where much of innovation comes from. Making that conversion to commercial grade is really a big challenge.
And we spoke a lot about regulation, and I think regulation is the other side of the coin for access, the other side being cost, but regulation, you have been exposed to MHRA and the FDA, and we talk a lot about long-term safety for gene therapy. What are the lessons that you have learned so far in terms of other developers can take home in order to improve their regulatory conversation with authorities?
I think, again, that's probably evolving. The long-term safety regulations are pretty arbitrary, I think, across the FDA and EMA and UK, that we have this 15 years of long-term follow-up. Even ideally, for people like myself and Don, we're really going to be following these patients lifelong anyway. We're going to be collecting that information. But again, I think with the regulatory innovation that's coming, across all of the different territories, we're moving to a place where long-term monitoring plans are going to be discussed up front, almost before the clinical trials already started. And that's almost, that's here now. We see that across all the different agencies.
And I guess, I would just add that for all the lentiviral trials at least, and I think also for AAV, there really have not been the sort of feared late complications that we're doing this 15 years follow-up. So it's an unfunded mandate. You can't get a grant for 15 years, and so we sort of cover it on a shoestring to follow these patients for 15 years. So I think that's something certainly that regulatory could make it a lot easier for companies where it's more expensive for them to do the follow-up. It's another burden. And so, I think 3 years, 5 years of follow-up is probably sufficient because there have not been late side effects from vectors to the present time.
And I think these are the lessons that regulators will adjust the regulation based on the learnings from the ongoing trials.
And I think probably maybe just one thing to mention again in that kind of regulatory landscape is that agencies are becoming more kind of amenable to like prior knowledge. And so when you can present data that shows that you've got like I said, like hundreds and thousands of safety data years for lentivirus, then if that can be considered as, you know, if appropriate in a new submission, then again that, you know, I think that could then adjust perhaps your monitoring plan for moving forwards.
And so for our ADA work, Claire treated the first patient with our lentiviral program in 2012, 14 years ago. And I know when we looked a couple of years ago, we had almost 500 years of patient follow-up, so we're probably around 600 now. And, you know, again, we just don't see anything late. You know, we get postcards on their first day of school and Christmas, and that's, you know, they don't need any disease-related issues. They're cured, basically. And so the follow-up is a bit perfunctory in a sense.
It's amazing the work that you have been doing, really, and you saved so many lives. And I can imagine how the families feel about this and how grateful they are. What do you think is the next big leap for rare diseases, and what is the thing that the field is not paying attention for? And maybe I'll start with Jen.
You know the answer.
Boy, do I feel put on the spot because I have about 5 answers that I'd like to fit into a short period of time. I mean, I can start with the second half of the question. You know, we're not paying attention to the diversity of the human genome and how, as we get more specific in the genetic engineering tools, what impact that lack of knowledge about human genome diversity is going to play, and whether or not those therapies actually have the market value that we think they could. You know, in terms of, I'm an agnostic about the tool. I feel like we need to have all the tools in the box because we haven't possibly predicted all of the diseases that we want to be able to treat. You know, we don't know how often we're going to be successful. I will add in, you know, in the regulatory reform conversation and everything else, you know, one of the things we have to think about is in the places where we value these drugs based on their benefit to patients and society. We're calling it, because of the underlying biology, a lifetime cure and pricing it as such, but we're really doing that with 2 to 5 years of follow-up data. And so we also need to revise the expectations and make it simpler and more affordable so that we can return income back to those companies to develop those things, et cetera. I think I would say, you know, this week at this ASGCT meeting, I'm most excited about all the different things like Rarity as a public benefit corporation that Don mentioned, all the different economic approaches people are taking to this problem. We've seen about a half a dozen. So there's the public benefit corporation approach, there's the commercial enterprise with a nonprofit subsidiary, there's a nonprofit with a wholly owned commercial subsidiary approach that we've seen come out. Plowshare Therapies would be an example of that. Caring Cross would be the example of the nonprofit with the commercial subsidiary. And we just had the Orphan Therapeutics Accelerator, you know, launch.
And academics.
And academic approaches, as Claire's working. I was getting there. You know, I think there's very good potential for more than one of these approaches to get good traction. You know, we're going to start to see this in the next few years when we start to see 1 or 2 of these approaches start being able to call their own shots, is the metric I'd like to be able to say. And that is going to create a tidal wave of change, in my opinion, because these problems are not unique to the cell and gene therapy space. They're applicable in so many settings. But in terms of innovation, in the scientific part, I'll go back to put my PhD hat.
Put money on it.
Yeah, put my PhD hat back on. Again, there's probably a million things I could say, but we are getting to a place where I really would like to see innovation in delivery. I think we're a little overcommitted to lipid nanoparticles right now, and I think we need to diversify a bit, and that is completely selfish as I'm developing my own approaches in that capacity. But I think getting to that place where instead of just jumping on board to the next thing and going all in on that one technology, we have to remember that we need all those tools in the toolbox because we don't know what's going to come down the pike. Does
Thanks, Jen. I think we need another podcast to go through some of these amazing points that you just brought up now. I will continue with Claire and Dawn if you would like to also add.
Sure. I mean, like if I'm gonna, I really hope that we're going to see some very positive results from in vivo delivery targeting stem cells. Because that would be I hope a great progress for accessibility and not having to have this complex and costly manufacture process, and that would simplify things. So I'm hopeful for that as a new technology. And I think in terms of I can't remember what it was. What are we not doing well? I'm not saying we're not doing it well, but I think that what we're getting better at is involving patients in the research design, the co-creation of things, and just recognizing and acknowledging their part in the journey. And I think also importantly, the regulatory agencies are doing that as well, and listening to the voices about, for example, outcome measures that really benefit or matter to patients rather than some arbitrary definition that somebody has decided. And so, I'm hoping to see again a lot more growth in that space. And you'll hear the meeting, and Terry Pirovalak is talking tomorrow. The patient-driven research results that we've seen have been phenomenal. And have taught us a lot what you can do in a short space of time with a reasonably little amount of money. But I personally don't feel that that's a way that we should be expecting things to go, because we can't put that burden on families and patients to do this at their time of need, so to speak. But they've definitely shown us what's possible. So that's my take.
I'll just jump in and echo there. I do think that as a field, we have benefited incredibly from tireless efforts of motivated parents and patients, and it is time for us to start making it easier for the next 100 diseases in line and not put the burden on the patient, but really, really take to heart the lessons we've learned.
Don?
Yeah, well, I certainly think that patient advocacy groups play such a major role in motivating us, for guiding us, raising funds for early phase work. So that's very important. In terms of technologies, I also think that, at least for the blood cell disease that I work on, in vivo, I'm hoping will be the wave of the future. Besides the complexity of the ex vivo cell manipulation, it currently involves getting a bone marrow transplant, being in the hospital for a month, getting high-dose chemotherapy. In vivo, ideally, you go down to the local drugstore, you get an injection, and you're treated. So it really could decrease costs in order of magnitude or more if it's effective.
These are all great topics actually that can fit into our special issue. And knowing how busy you are, I'm very grateful that you made the time and agreed to serve as guest editor for this special issue. So maybe just as some closing thoughts for our audience, you would like to share what kind of questions you encourage our authors to tackle or submit to this special issue. What would you like to see in it?
I would like to see, obviously, rare disease research, but I'd really like to see some of our early and mid-career researchers, putting in to the journal. I think we need to stimulate the next generation of researchers, and I think molecular therapy is very open to supporting that.
And I hope the issue sort of represents the diversity of the field of gene therapy, from CAR T cells, from AAV, from lentiviruses, for non-viral. There's so much going on. And so hopefully we can represent all that as applied to rare diseases.
I mean, I think I would love to see a diverse set of authors contributing just at the heart of their research and or regulatory and or economic burdens. The sooner we start talking to each other about all of the things happening that are impacting rare disease, cell and gene therapy moving forward, the sooner we'll get to innovating in solution, and that will be the inspiration for those next generations that Claire mentioned.
Thank you. I'm sure Terry would have joined you as well in the same enthusiasm, but he had to leave for a meeting. But we are very grateful for you and for your time. I'm sure molecular therapy advances will benefit a lot from your experience and your dedication to the field. So thank you again. Thank you for your time today. And I think that's it for our podcast. We appreciate it.
Thank you very much.